Miss Jody here with another video for biology that meets on Tuesdays from 1 to 3:30. Hopefully you're all doing well and we are moving along here in our lessons. I'm just doing short videos. I might have to make a few extra because we're creeping along in these chapters. I'll see what amount of time I can get in later in this week. Maybe I'll make another one too, so we have a couple videos. But our last video was introducing the kingdom Animalia, moving on into all of our classification. Now don't forget when you're taking your notes to put them on a half sheet, and that way I can glue them in and just write the title on the back so we can keep the journals up. Hopefully we'll be back together a little in May, I'm hoping, but who knows. But I would like to get back together because we won't be able to do any of our dissections, and that's a big part of the biology study. And so I definitely want us to get back together at some point and do some of that stuff. So hopefully we can just get a lot of these notes done during this time. So I know it's a little boring and not super exciting, but it is what it is, right? So with Kingdom Animalia, starting with this particular category of the kingdoms, we were looking at, okay, what are the characteristics of animals? And so certain things will get you in this category of being an animal. And then once you get in that category of being an animal, then we've got to put you into lower levels of classification. So into then phylum, class, order, family, genus, and species, and then try to figure out by your characteristics where you fit best in all of those particular categories. And of course we talked about Kingdom Animalia being divided up with one big category of whether you are an invertebrate or you are vertebrate. So that's something we're going to take a look at now that we move into our first category. And last week when I sent you this on the germ layers, there was a second sheet that was attached to it, which was this one, because I try to do in half sheets so you don't waste too much paper and ink and all that. And this is the one we're going to use today on studying the sponges. So in the sponge world, and I love the sponges, and when I send you some videos to look at some of these creatures, take the time to look at them. And even though they might seem a little juvenile because they are younger version videos, they are just so great in their illustration of how it works in the sponge world, specifically the one on the sponges that I always watch in all my classes anytime I teach on the phylum Porifera. All right, so sponges, they are invertebrates with no germ layers. So remember we learned about the germ layers at our last video. No nervous system, digestive system, or circulatory system. And so they would be thought of as more simple creatures, right? But they actually are still very complex, and obviously being in Kingdom Animalia, you're of the more complex. And then we look at them and we can categorize them as sessile. Sessile means they're kind of attached to one area. Like if they're on the bottom of like the ocean, they would be, or like a bottom layer, they would be what we call benthic, bottom dwellers. But if they're sessile, you can be benthic and still move around. Or you can be benthic and you're sessile, you are actually attached to something, you don't move around. So attached to the rocks and they stay in one place. Symmetric, and we learned about symmetric side of things in our last video. They used to be thought of as plants, and it makes sense because when you do look at the sponges, and the video I send you that's looking at the sponges, it gives you some insight back when the sponge divers were getting the sponges out of the ground or cutting them off and not really realizing what they were doing, because sponges are huge filter feeders and they keep the ocean clean through that process of filtering. And so by killing off these sponges was causing damage to the ecosystem. So they used to be thought as plants, like no big deal, and just cut them out. They are eukaryotic, multicellular, and heterotrophs. Obviously, you got to be that to get into the category of animals, and that's why instead of being in plants, they started saying, oh, okay, so they're heterotrophs, they're eaters, they're not going to be making their own, though they will be in relationship with algae. They will have a symbiotic relationship, some of them with algae, but they themselves are not autotrophs. So the phylum, the big phylum that these sponges go under is Porifera, and the Porifera means pore bearers, and it makes sense because these sponges have all these pores, and these pores are where they bring in the water, and it gets filtered through, and they pick out, they engulf the debris and the plankton that comes through those pores. So they can be really, really small sponges. I have these little tiny, tiny sponges in a little container. I don't know if they're on the counter over here. Oh yeah, I actually have one. I knew I saw one over there. Let me grab it. And for any of you that missing the squeaky picnic tables at the Science Center, then me getting up and down should get you a squeak or so, hopefully. Because we know we miss those little squeaky things up at the upstairs lab. But this is a little container that has a little tiny, it's hard to see, but has a little sponge in it. And so they can be really, really small and or really, really large. And the video I'll send you will show you some of the big ones that are really super cool. And it's really cool to watch how the water comes in through the pores and then spits out at the top when these scientists put dye in the water. You can actually watch that process of filtering. I love that. That video never gets old. Some of you that have been in my biology classes before will have already seen it, but just watch it again and just enjoy it to the depths of your soul like Miss Jody does, okay? All right, so they can be small to all the way large. They can be marine or freshwater. So there's about 10,000 species of these sponges. So they reproduce sexually and asexually. So anytime we're talking about a sponge, all right, or any of the categories will always be saying, okay, what is their reproduction? Because obviously we're in biology and that's something that we have to pay attention to. And so with the sponges, they are let me see, I'm going to say this wrong again. Hermaphroditic, yeah. Hermaphroditic. I always say it wrong, but you know what it is. Hermaphroditic, no. Aphrodite. Aphrodite, yeah. Hermaphroditic. Oh, whatever. Anyways, that's just giving you a little taste of, I miss Miss Jody doesn't know how to pronounce anything. There you go. There it is, if that's what you're missing. All right. So that means you have male and female parts. And so you're able to release sperm into the ocean, and then the eggs that stay attached to the mother's sponge until they hatch, and then they release larvae. And then once they release as larvae, they have the ability of motion, so they can move around to the place where they're going to end up settling down. So it can be in this way, where whether it's the particular releasing of the male and then a different sponge receiving that. And so that will be reproducing sexually, okay? Now, if they're reproducing asexually, and my book is getting a little fat here, so let me move it up here so it's a little easier to see, is when we're going to see the budding. And we've talked about this a lot with different organisms. There's a budding. It's almost like getting a new limb on a tree, you know, the budding process. So it can go in, it can reproduce asexually like that, or for the gemmules, which can be released in that, which is still going to be genetically the same for an asexual reproduction. And so there's no mixing up on these two compared to this mixing up between the sperm being released in the ocean and whichever sponge mother receives the sperm in her eggs, then you've got a mixing up of genetic code. All right, now with the sponges, they're filter feeders. And like I said, a filter feeder is an aquatic animal that filters out plankton or nutrients suspended in the water. So they are very important to the ecosystem, and they found that out the hard way. And now many people like to use synthetic sponges instead of killing the natural sponges, though there is still sponge harvesting going on, but not like it used to. And then there's also the fact that, like I said, the symbiotic relationship with algae. So sometimes, well, a lot of the time, the algae is living in the structure of the sponge, and so therefore giving the sponge some extra nutrients by in that filtering process as it gets pulled through. And so the sponge is providing safety while the algae is providing extra nutrients. Now, when we look at the diagram is to help us figure out what's going on here with the sponges. And the sponge world, I had the diagram that I sent you, just giving you some categories of sponges, because they're not all the same, but they have similar characteristics. So this is representing more of the simplest form, and this is kind of like the middle form. So this is the ascon, and this sicon, and then leucon. So that's more of a simple form. This is kind of the middle form, and this would be the most complex form. But they're all going to be working the same way, okay? So what happens with the sponge is we have basically they are tubes, just like most of the animals. There's some sort of tube side to everything, and so they're tubes, but they have only one open end, meaning the water's going to come in and then it's going to be filtered out, but like a normal tube, you'd go in and out both ways or whatever. So with the sponges, how it's going to work is the water going to come in through these pores. So this blue crayon is going to represent my water. And so as the water's coming in through the pores, and that's in all of these versions. So the water's being sucked in a sense. The way it's being sucked in is the lining of the inside here, and this inside is called an osculum. The lining has these little collar cells, and these little collar cells, you'll see a picture in our book, they have these little flagella, and these little flagella kind of keep whip-like behavior, which creates this current. So all of these little cells lining the osculum will create this current, and as this current is being created and they're whipping it up, then they're going to be bringing the water in from the outside, and it'll be flowing out the exit, which will be where the waste product, anything they don't end up using, will flow out. And so it'll eventually make this current because of these little collar cells and flow through. Now, during this time, as there are things suspended in the water, like plankton, which plankton is a category. Sometimes people think of plankton and they think of these little microscopic organisms. That is true. But even the big lion jelly sea creature, what's sometimes called lion or the jellyfish, which I don't like to use the word fish because really, the jellies are not fish. They don't meet the qualifications to be in the fish category. So I just call them jellies. So the jellies, the big giant lion manes jelly, is considered plankton. So plankton is a category, meaning that it's anything that cannot overcome the current. So if you're a floater, if you're not a true swimmer who can swim out of the current and direct yourself on your own, you are considered plankton. So anything floating in this water is going to be plankton. Now, of course, these sponges aren't going to suck in a big old giant jelly, right? But anything that can get through these pores that is suspended in this water, which will be any of the plankton, the living organisms that are floating, and or the nutrients that some of the minerals and such that might be floating in the water also that aren't necessarily living, and there'll be debris or whatever, they can move through and come through the pores, and then these collar cells can engulf them. So imagine all these little things start coming through, and as they're coming through with the flow of the current that the collar cells are making, then there's an opportunity for feeding. And so what happens is these collar cells that are flagellated line the inside of the sponge to create the current, right? Then the collar cells engulf the food particles through phagocytosis. So remember phagocytosis? Phagocytosis. Okay. is when phago is going to be the engulfing, and cytosis is the cell. The cell itself is doing the engulfing. Remember, we saw that a lot with the the protozoans when we were looking at the protozoa. So they will pull it in and kind of engulf it. And this is the process we call intracellular digestion. So intracellular digestion is, since you don't have a digestive system, how are you going to get your nutrients? Well, your cells are going to do the digesting. And so they do the digestion so that they have what they need. And what happens, and it's kind of cool, is they partially digest the food, and send it, and then it ends up going to other cells to finish the digestion. So they only digest some of it, and then it moves on to the next cell, in that process of that current. And so that way, they don't really need a circulatory system to like bring nutrients all throughout their particular bodies. The water they're living in is sort of the transport system that bring the nutrients, and to bring the debris out, the byproducts, whatever they don't need, can be brought out of the cell and then out through the current. So this allows nutrients to spread in the sponge. And then the waste products are ejected through the osculum, the middle part. Now, one thing I missed, I see here, is that the walls of the walls of the actual organism are supported by these little needle-like structures called spicules. I try to draw it. It's better drawing in your book. But they're embedded in this outside part of the sponge, the exterior part of the sponge. They're actually underneath it, of course. So they're still in the interior, but it's pushing out the structure on the outside that makes them kind of firmer. So that's kind of cool with the sponges. So the sponge is a very firm organism that is a filter feeder that's going to be very beneficial to the ecosystem. And then our next one, the Cnidarians. Now the Cnidarians are one of my favorites. They're just so beautiful. So of course we got to do some sort of craft or something, because I don't want to have just these boring journals. I just it will just break my heart. So we got to put a few like crafts. I know we got a lot of diagrams and stuff in here, but we got to do something beautiful, right? Like, remember this little beautiful corn that you absolutely loved when we were doing population genetics, and everyone was like, can I do another corn? I love this corn project, Miss Jody. That's all sarcasm. I know you hated it and me making you roll little corns, but isn't it so cute? Wow. Okay, so we have to do something for the Cnidarians. And since they are very beautiful animals, I'm going to just do a little something that you can do at home so we can at least have something for this. Okay? All right, so the Cnidarians, they are invertebrates with 2 germ layers. They are, instead of being rigid like the sponges, they're softer creatures. And they're symmetric instead of being asymmetric like the sponges. So they have a symmetry to them. And then in the category of Cnidarians, there's all the jellies, which are normally, like I said, called jellyfish. But since they are not true fish, they don't have fins and gills, and they don't meet the classification to be called a true fish, then we need to keep them as jellies. So jellies, all the corals, and all the sea anemones. So what's cool about these guys is their nervous system, it doesn't create like a true brain, but they have this what's called a nerve net. So they do show signs of this nervous system being able to have all the sensory stuff that we want to start seeing in these more complex animals as we move up. So they will have a sort of like a nervous system, with no true brain. Excretion and respiration. So whenever you have your excretion process, you're like, oh, well, you got to have some kind of system to get that stuff out. Or respiration, you got to have some kind of system to bring in your oxygen. But they don't have a full system like what we would see with most animals, or like higher level animals, but they're carried out at the cellular level. So that means the cells themselves, like we were seeing with the cells of the sponges engulfing the food and creating and doing the digestion, intercellular digestion, we now have intercellular excretion and intercellular respiration. So the cells are doing it at the cellular level. So when we look at the Cnidarians, one thing we notice is that they rely on water for support. And we talked about having this skeletal being an endoskeleton or an exoskeleton, and that there's the hydrostatic skeleton. That's where the water pushing out from the inside of an organism out to the outside and being based in water, creates that. So obviously the jellies have the hydrostatic. The corals actually end up making an endoskeleton. No, I'm sorry, not exoskeleton with the what's it called? Oh, the calcium carbonate. Oh, I did write it here. Corals excrete calcium carbonate to produce exoskeletons for extra protection. So they still rely on the water for support, but they produce this extra harder calcium carbonate to have extra support. I mean, extra protection. So they're firmer, of course, and you know that with the corals. Okay, all of these guys, the jellies, the corals, and the sea anemones, they're named this Cnidarian name, they're named for their stinging cells. Now, stinging cells, the big bulk of the stinging cell is the cnidocyte, but when you learn about it, there's these little parts inside called nematocyst, and that's what I always focus on is the nematocyst. And these are specialized cells in like the tentacles or whatever of the organism. All of these three, the jellies, the corals, and the sea anemones have these. So they're like these specialized cells that contain barbed or venomous coiled threads that can be projected in self-defense or to capture prey. So there are these special little cells that have these little barbs or like these little coiled threads where it can, well, actually the coiled little thread kind of makes it like a springy thing, and then it shoots out, and you'll see a picture in our book shows the process of it shooting out. And so basically when you touch a jelly and you get that stinging, what's happening is you touched it, and the part of the cell is kind of like a trigger, and it springs out because you pushed on it. It springs out and it shoots that toxin, that venom, into your skin through those little injectors. And if you remember anything with biology, whenever somebody's venomous, that means they have a way that they've injected it into you. Poison would be like absorbed through the skin or ingested through the mouth, but venomous would mean something injected it into you. So these nematocysts fit right with the venomous word because they actually have these little barbs that inject it into you. And then the whole word of the Cnidarian cnidocyte, okay, there we go. I was going to pronounce it wrong again. Cnidocyte is the whole package of that with the nematocyst. So an explosive cell containing one giant secretory organelle. And so it's this cell as a full package with the nematocyst to create this big defense ability for the Cnidarians, and also for them to capture their prey, to put the toxin into their prey so they can get them. So with reproduction is asexually and sexually, like the sponges. So the difference is, though, in asexually, which I did the asexually one first on this one, they divide in half. They can divide in half and recreate new, or even the budding process that we talked about with the sponges and many other organisms. And or there's also the sexual reproduction, which is the same thing. In a sense, with the sponges, you've got the sperm and you've got the egg. But the difference is the Cnidarians are not both male and female. They are typically either male or a female. They don't have both parts. So the sperm will be released in the ocean and will eventually fertilize the ova that was released by the female. And so in this Cnidarian world, the jellies fit. And so, and I'm going to send you some videos on the jellies and the corals and stuff, because it's pretty fascinating, all these unique invertebrates and how they fit into our biology. So much amazing, complex design. I mean, I wish we were together and we could talk a lot about it, because it's just phenomenal. All right, so I thought we would do like a little craft here, and I'm going to send you some videos on like drawing jellies and all that, and you can pick whichever one you want to do. What I was going to do, because I'm trying to think of limited supplies here that I have, is I'm just going to cut little pieces of tissue paper in squares, and I'm going to just do one of my favorite things, which is always simple and cheap, because I have the supplies, is like the tissue paper art. And so that way I have something for my book. So I just cut some little squares of tissue paper, but again, I'm going to send you videos on drawing and stuff, so you can do whatever you want, but do something so that we have something looking fantabulous. And then I've got to put in some of my Faduzzle, because it wouldn't even be right in life if I didn't have that on about 5 of our projects, right? It wouldn't even be right. So I'm just gonna cut these little guys, and then so they're not too long, and then I'll just have them dangling on my blue paper. So I'm gonna just have my little tentacles with my little nematicist on there, and then put some glue water on and put some little green tissue paper. Easy peasy pudding pie, or whatever. I know I always say that one wrong too. Easy peasy pudding pie or something. I don't know how it goes. All right, so I got a couple of these little strings, and then that's going to be my tentacles. And then I got some little, put a little glue in a little container here, put a little water on it, slap that right all over the place here. These will stick to it for my little jelly, and then stick on some tissue paper. Now I made it in squares, so it's kind of abstract in the sense that jellies are not going to be square-like, but hey, that's just my method of my tear art. Usually the things I like to do is always like this kind of stuff. Easy. And so it'll be kind of boxy. But hey, the box jellies are in this category too, so it could be fitting. And like I said, I'll send you some videos. I'm gonna try to draw a little umbrella-like. Oh, I did forget to note on there the life cycle, how they will, once they do reproduce, you can notice it in the book, the different shapes, the medusa of the Cnidarian and or the polyp. So this is how I remember it. And again, this is all going to be on your book, but I always think of Medusa is like the Medusa with the long hair and all that. So this form right here that I'm showing you is the Medusa form, because they have they're floating around, they've got all this hanging from them. But some of the Cnidarians, when they're smaller, will be in the polyp form, and they'll kind of be this way, and then they'll attach to the bottom of the ocean or wherever, not the bottom of the ocean, but the bottom of where they are in the water, and they will be sessile for a while and then produce the Medusa forms to be budded off or whatever. And so there's a whole life cycle in your book that can give you a better illustration of that. Maybe I'll put a video in for that one too, just so I don't forget that one, since normally I would have probably, for this part next to the Cnidarians, I probably would have put the life cycle, so we'd have the diagram of it. But then I just wanted to have something artsy in our journal, since we haven't been able to do anything fun since we've been separated. Like I said, hopefully we'll get back together, because I really don't want to have a whole year or a year of biology that doesn't get to do all the dissections. That would be just horrific. Now, some of you that have already done biology are gonna a first level biology, you're gonna go, yay, I don't want to do dissections. Some of you, but some of you would be like missing out if you don't get to do a dissection. So I'm hoping that we can get together and do all of our dissections. But anyway, so how's it look? Just fabulous. So there's my little jelly. I'm gonna stick them in there so that I have something to not make my journal look so boring. Okay. All right, so I might later this week make another video, since we have a lot of topics in each chapter. I can't really spend 3 weeks on a chapter. We're supposed to be doing a chapter per week, but you know how this book is filled with so much stuff. I don't know that I could get that done. But we definitely can get a little farther. The next one's on worms, the category of worms, and then what's the other one? Oh, then mollusks are next, and then Echinodermatas, like the sea stars and all them. All right, I'll see you next time.